Hey guys, I want to do a really quick crash course on amino acids. I want to walk you through this guy right here. Here is our basic skeletal structure for an amino acid. And there are 4 parts to it. And in amino acids, ooh, that's a horrible color. Let me get a better one. Amino acids, you'll have the N terminus, you'll have the C terminus, and you'll have the R group, pretty obvious there. Then you'll have the alpha carbon, which is the central carbon in an amino acid. The variable part of an amino acid will be the R group. The rest for every amino acid will stay the same. In every amino acid, well, there's one exception, but you don't need to know that now, is you'll have an N terminus and a C terminus. Okay. Something to remember is when you're talking about amino acids and then proteins, both will actually technically have an N terminus and a C terminus. Here, for obvious reasons, this is the N and then this is the C. But also when you have a polypeptide, you'll start building it amino acid by amino acid, and you'll start to see a pattern here, right? So NCC, NCC, down a skeletal backbone. That's just following this pattern. NCC, when you add another amino acid to that, it'll continue the same pattern. So this is your polypeptide backbone, and this will be your N terminus and your C terminus. Same thing. So both polypeptides and an amino acid by itself can have the N terminus and C terminus. One thing I want to talk about is the functional group, the R group, which are synonymous in this case. Your R group is your opportunity for variability in an amino acid, and that is what differentiates one from another. Now, some amino acids will have acidic or basic or aromatic or hydrophilic, etc. But one thing we really want to take care is when we use these descriptors for an amino acid. Now, we will always be able to say that the functional group is one of these. We can say that the R group is hydrophobic. We can say that it's hydrophilic, acidic, basic, aromatic, sulfur containing. But we can't always say that the whole amino acid is going to be hydrophobic, hydrophilic, for example, because actually this whole molecule is going to be hydrophilic. Because you have the nitrogen here, which is a pretty polar molecule. You have oxygen here, which is very polar molecules. And those will cause a lot of hydrophilicity. So you're not really going to say that the whole thing is hydrophobic because you have very hydrophilic regions. So you've got to really be careful using these descriptors when you talk about the entire amino acid. But you will always be able to use them when you describe the R group. So that's something to watch out for. Now, an amino acid, I'll abbreviate that with AA, is known as a monomer. Now, there are lots of types of monomers. An amino acid is always a monomer, but a monomer is not always an amino acid. You can have glucose, for example, is also a monomer. And a monomer is just a name for the simplest unit of something bigger, right? So in a protein, your simplest unit in a protein would be an amino acid, basically. And when you increase the number of amino acids, you go to a polymer, right? And the protein, well, actually, technically, you can have a peptide or polypeptide, either one will do, is a polymer. A protein is a polymer, and I'll get into how to differentiate those two in the next video on proteins. And yeah, so these are the polymers, the polymeric units of amino acids. So amino acid is the monomer, and the protein is the polymer. If you had just 2 amino acids, then you would have a dimer. Okay, that's just when you have 2. So just some really basic terminology. Hope this clears up something for you guys, and thanks for watching, and keep studying hard.